All right, hello everyone and welcome. In this chapter we're going to be talking about enzymes, macromolecules that catalyze chemical reactions with very high selectivity and specificity. If we think about biological processes in living systems, most are chemical reactions, and those chemical reactions are catalyzed by enzymes, or the majority of those chemical reactions are catalyzed by enzymes. For instance, enzymes catalyze all aspects of cell metabolism. So what properties of enzymes allow them to control a chemical reaction, and specifically control the specificity of a chemical reaction, meaning does a given enzyme bind to any sort of substrate, or can it bind to a number of different substrates? And just in case you're not familiar with the term, so what an enzyme does, it's a catalyst, right? It converts a reactant into product, right? So the enzyme is really a catalyst, right? And what it does, it's converting a reactant into product, right? So there are specific type of terms that we use. We typically abbreviate the enzyme as E, right? And the reactant is really our substrate, and the product is P. So we say that our substrate, S, right, is converted into product. The substrate, which is the reactant, is converted into product, and the enzyme is necessary to convert that particular substrate into product. Now, the substrate binds the enzyme at what we call its active site, and depending on properties within the active site of the enzyme, that dictates what type of substrates bind to the enzyme. All right. And that's what we also refer to the specificity of an enzyme. Does it bind any type of substrate at its active site, or does it specifically only bind to specific type of substrates? All right. Some enzymes contain other metal ions or cofactors. One example is what we just saw right here with carbonic anhydrase and the active site that has a metal ion, a zinc ion, which is essential for the activity of the enzyme. But we're going to be talking about some examples of those as well. All right. So how can we compare a chemical reaction catalyzed by an enzyme versus an uncatalyzed reaction in the absence of an enzyme? Well, the turnover of a substrate into product in the presence of a given enzyme increases the reaction rate, right? So the efficiency of that reaction is very high in the presence of an enzyme, right? If you think about organic reactions, those are typically carried out in organic solvent, whereas in enzymes can catalyze reaction under neutral pH and under physiological conditions, basically, right? And they have quite specificity towards what type of substrate a given enzyme binds, or even what type of reactions it catalyzes, right? And unlike organic chemistry, if you think about those type of reactions where there's a number of side products or possible products, an enzyme can catalyze the reaction or a given reaction without any side product. So it's very efficient, right? The specificity is high and the efficiency is high because of the rate. Does it recognize different chiral centers or stereo differences in stereospecificity? It does. So it's very regioselective and stereospecific as well, right? And does anything turn off the enzyme, or does it always, once it binds to the substrate, turn over into product? So an enzyme is actually regulated by different substances that can bind to the enzyme and alter that rate of the reaction, or in other words, control it, right? On a later chapter, we're going to be talking about allosteric control, covalent modification, and how variation of the enzyme that is synthesized can also help regulate the activity of that particular enzyme. All right, so how exactly do we classify enzymes? Well, the very first enzyme that was named, it was like in 1833, I believe, and the name ended in ASE. And years later, a microbiologist and a chemist decided why not name all the enzymes and let them end in ASE. So at the end of an enzyme, you'll always notice that the name ends in ASE, right? And the enzyme is really the name of the enzyme is referring to what type of reaction it catalyzes. Not all the time, but quite of the times, right? Urease, for instance, catalyzes the hydrolysis of urea, and hence the name urease. All right. Catechol oxidase is another example of a name of an enzyme, and it's an enzyme that specifically catalyzes the oxidation of catechol into its orthoquinone products. Right. Alcohol dehydrogenase is an enzyme that catalyzes the oxidation of primary and secondary alcohols to their corresponding aldehydes or ketones by removing a hydrogen. It was a reaction that you covered in Organic Chemistry 1, right? But we weren't using an enzyme, we were using a reagent in that case. You remember what reagent it was? All right. Does the name of the enzyme always indicate what type of reaction it catalyzes? Not really, right? The name of some enzymes really give you no clue about the function of that particular enzyme. And catalase is one example. It mediates the dismutation of peroxide into water and oxygen. This should be a small 2, a subscript, of course. All right, so if the name ends in A and ASE, we're talking or referring to an enzyme. All right, so how can we classify enzymes in a larger scale? Well, they're classified as one of 6 classifications. So enzymes belong to one of those classifications, right? They're known as, they belong to oxidoreductases if the enzyme catalyzes oxidation reduction reactions, right? Enzymes that belong to transferases are enzymes that catalyze the transfer of a functional group, right? Enzymes that belong to the class hydrolases are enzymes that catalyze hydrolysis reactions, right? Lyases are enzymes that catalyze the elimination to form a double bond. Isomerases, that just like the name imply, are enzymes that catalyze the isomerization, right? Taking the molecule from one isomer, to another, right? They differ in the way they're connected. And finally, ligases is a class of enzymes that catalyze the formation of a bond in the presence of an ATP, or really coupled to the hydrolysis of ATP, and that is the source of energy for that particular reaction. All right. Okay, so those are the larger classes of enzymes. So all enzymes will belong to one of those 6 classes of the enzymes, depending on the actual type of reaction that particular enzyme catalyzes. Okay, so when an enzyme converts a given reactant or a given substrate into product, the substrate binds to the enzyme at its active site, right? So you have a substrate, and that is what's being converted into product, and we have an enzyme, and the active site of the enzyme, this is really an example, or really right here, is an example of the active site, right? The active site of the enzyme is where the substrate binds, right? And then the substrate is converted into product. So we can actually describe this slightly differently in that we have the enzyme initially, right? and the enzyme is reacting with the substrate, or the substrate is reacting with the enzyme. And then we have an equilibrium, and then what we form is the enzyme substrate complex, right? What we refer to as the ES, the enzyme substrate complex, right? The ES. And then what happens later is now the enzyme, is not consumed in the reaction. The enzyme simply is where the substrate binds. You have an enzyme substrate complex, and then you have the enzyme once again, and the substrate is converted into product, right? So the overall enzyme is unaffected by the reaction, or is not changed. It might undergo conformational changes while the reaction is taking place, but the state of the enzyme initially and at the final ends up being the same once the reaction is catalyzed, right? Or once the enzyme substrate complex is turned over into product. All right, so if a given enzyme has an active site, and that's just the folded protein will fold in such a way that there's this hydrophobic part of the protein, or somewhere within the protein there is what we call the active site, and that's where the substrate binds to the enzyme. So it is regulated by both geometric and electronic complementary, so meaning that the substrate has to perfectly fit within the active site, right? It has to have groups within the substrate that will be stabilized by the side chain of amino acids, or even the peptide backbone within the enzyme active site, right? Those are typically non-covalent interactions, right? Sometimes what we get is an induced fit, right? Meaning once the substrate binds to the enzyme will undergo some sort of conformational changes, so that its affinity to the substrate becomes higher, right? So you have an enzyme at a state 1, the enzyme might undergo conformational changes and become E2, but once the substrate is released, the enzyme goes back to its original conformation, right? And that's what we call induced fit, right? Perfect. All right, and we know that amino acids found in all proteins are really L amino acids, right? And if you think about citrase versus isocitrase and it reacting with the active site of the enzyme, there's only one way for it to bind to the enzyme. Although it has 2 carboxylic acid groups, there's only one specific way where the actual substrate can interact with the enzyme. So it's highly specific, right? And that's what we call or refer to as stereospecificity. All right, so what about geometric specificity? That has to do with the active site itself and the substrate itself. Does it fit very well within the active site? Does it matter if there's small variation within the substrate or not? Then we're talking about the geometric specificity. And enzymes vary. Most catalyze only small range of substrates, right? So a given enzyme might only catalyze few different substrates. Some, and the efficiency then is different, meaning the rate is different, right? So if you have another substrate that can still bind to that particular enzyme, form an enzyme substrate complex, and then that substrate is converted into product, the rate of one reaction using substrate S versus substrate S1, the rate is not the same. And that's what we call different efficiency, right? One reaction might be faster than another reaction, or more efficient. All right. So their efficiency is different. And some enzymes catalyze a number of different, or the turnover of a number of different substrates, right? And sometimes we can describe that as a preference, right? And digestive enzymes are such example, right? And some are not very specific at all, so they can catalyze a number of different reactions. If we talked about a chymotrypsin in a different chapter where it catalyzes or cleaves a peptide bond right after aromatic side chains, phenylalanine, tryptophan, tyrosine, right? But if you recall, it also had some amino acids that it can cleave after, but a much smaller rate. That would be an example of different efficiency, right? An example of different types of reaction is, in one case, chymotrypsin catalyzes the cleavage of a peptide bond. In another reaction, chymotrypsin can catalyze the cleavage of an ester bond, right? So that's an example of different reactions, right? Okay, so what else are present in enzymes? There's the active site of an enzyme, and it differs really by the alignment of amino acids, or what amino acids are present at the active site, and how are they stabilized. But some enzymes have cofactors, right? And cofactors are molecules that are essential for the function of the enzyme. They are either metal ions, copper, iron, zinc are all examples. We're gonna be talking about enzymes that have copper, iron, and zinc at the active site. Sometimes if you have a metal ion that has similar, you know, in terms of electronics, if you have a metal ion that is similar, but you replace one metal ion with the other, it can render one enzyme inactive, meaning there are metal or enzymes that have copper at the active site, and if you go in and you replace copper by zinc, that reaction that the enzyme catalyzes might become inactive or might slow down quite a bit. All right, so there's metal ions. Some are coenzymes, and coenzymes is really not referring to another enzymes, but they function as co-substrates, right? So the term coenzyme is really kind of, those coenzymes are functioning as co-substrates. They will initially associate with the enzyme, right? But they're not permanently associated with the enzyme, not necessarily, right? They're either co-substrates or prosthetic groups. We've talked about hemoglobin at this point, and how it has a prosthetic group that is permanently associated with the enzyme, right? But co-substrates, right, are only associated with the enzyme when the enzyme is catalyzing a given reaction, and then they dissociate from the enzyme. All right, an example is NAD+, right? ADH is referring to alcohol dehydrogenase, which converts primary alcohol into aldehydes, right? And the example here is ethanol into acetaldehyde, where it's really simply acting as an oxidizing agent. But it binds to the enzyme or interacts with the enzyme, right? And hence it is known as a coenzyme, right? All right, and it functions as an electron carrier, NAD+, and NADP+, right? And it's also involved in reduction reaction or reduction addition of electrons and the transfer of two hydrogen atoms. And if you think about the reactive part of NAD+ or NADP+, right? We didn't really talk about nucleotides yet, but you have a nitrogenous base, you have the sugar, you have the phosphate, and then it's connected to a D-ribose in this case, and then to a nicotinamide, right? And it's a electron carrier in the form of a hydrogen with a lone, with its one and only electron. So if you take a look, you have a nitrogen with a positive charge. You have a cyclic molecule, it's fully conjugated, right? So what is happening is that you could move those 2 electrons to the nitrogen. That would create a positive charge right here, but you have a double bond right here. You could move those electrons right there, and then that hydride can attack right here. All right, so you have the oxidized form NAD, P plus in this case, into NADPH. Right? So it is an electron carrier. So if a given enzyme has a prosthetic groups attached to it, that prosthetic group is permanently associated with a protein. It's either covalently or non-covalently, but it's permanently associated, or it's covalently associated with the protein. And an example is the heme group of hemoglobin that we talked about. So if an enzyme requires a coenzyme, right, for its function, in the absence of the coenzyme, that particular enzyme is known as an apoenzyme, right? In the presence of its cofactor or coenzyme, it is known as a holoenzyme, right? The term apoenzyme is used more often than a holoenzyme, because when we're referring to the enzyme, we're assuming, or not assuming, but anyways, the term apoenzyme is used quite often to indicate that that particular enzyme, its coenzyme is not associated with it at that particular reaction or structure that we're looking at, right? So an apoenzyme is referring to the enzyme without its coenzyme, right? And again, coenzymes bind, or co-substrates bind to the protein and then dissociate, right? So they're not permanently associated with a protein, unlike, or an enzyme, unlike prosthetic groups. Okay, so energy diagrams, we're looking at the free energy change versus the reaction coordinate. So what's happening during that particular chemical reaction. And you have 2 hydrogens, hydrogen A and hydrogen B in this case, and you have hydrogen C. It's just for an example to demonstrate what is happening. You have the free energy of activation, and then we have the transition state, and if you recall, that's where bonds are formed and bonds are broken, right? You have the initial reactants, and then you have the transition state, and at the transition state, that is where bonds are formed and bonds are broken, right? This is what it's, I'm not sure that you could see it. HB plus HC. And then here they're swapping, so meaning that this bond is breaking and this bond is forming. But that transition state, that's the high energy molecule where bonds are forming and bonds are breaking. And if the energy initially is very similar to later, or later has a slightly higher energy, the reaction overall is endergonic or not favorable. However, if there is a net release in free energy, then we know that reaction is favorable, right? And if you recall, we refer to that as the energy of activation, right? That energy barrier that that particular reactants need to overcome in order to turn over into products. Ta-da-da. I actually have them labeled. And again, I'm not sure that you see it, but all right. So you have the transition state, you have the energy of activation, and you have the starting material, right? So what's the difference between those 2 energy diagrams? We saw, or we learned in organic chemistry, that an energy diagram that is only one transition state, meaning that the entire reaction takes place at one, in one step, like an SN2. All right, well, if you take a look at an energy diagram and then you see multiple transition state, then you know that Oh, all right, going too fast. Multiple transition state, then you know that it's a multi-step reaction, right? You start with reactant A. I'm going to follow the red. And then it goes up. That's the transition state. And then you have a reaction intermediate. It's a product formed in the process, but then consumed later in the reaction. And then you have a transition state, and then that product is turned over into reactant. In this case, what is determining the overall reaction rate? The first step of the reaction or the second step, right? It would be the second step, the one with the higher energy of activation, right? So in this case, the second step is what we call the rate determining step. Well, what about the other type? All right, A, and then it goes through an energy of activation, right? And then another. So in the second diagram, the first step is the slowest step of the reaction, or the rate determining step of the reaction. All right, are those catalyzing the same reactant into product? Yeah, but they're taking different pathways, different mechanisms, right? So it's the one with the highest energy of activation that is the rate determining steps. So what about when we have an enzyme, right? What enzymes do, we learned in general chemistry that it lowers the overall energy of activation, but what it really does is that particular reaction will take an alternative pathway, right? And that's one example. In the absence of an enzyme, it was two-step, but the second step is the rate determining step. It's the slowest step. And assume 2 is in the presence of an enzyme, the blue line, where the first step is the rate determining step, right? So it takes a different mechanism, right? And that's why it lowers the energy of activation. So that's just 2 examples of the uncatalyzed with a higher energy of activation versus the catalyzed reaction with a lower energy of activation, right? So what exactly is the enzyme doing to the given substrate that the enzyme catalyzed reaction has a much smaller free energy of activation, right? It really has to do with how the enzyme stabilizes the transition state, the ES complex, right? Which reduces the free energy change, makes it smaller, and makes that reaction faster, right? So an enzyme stabilizes the enzyme substrate complex, right? Which makes that delta, or the energy of activation, smaller. All right. And that really also has to do with the specificity of an enzyme to a given substrate, right? Can it stabilize the enzyme substrate complex? In fact, at a later chapter, we're going to be talking about inhibitors. And if an inhibitor binds to the substrate, to the enzyme instead of the substrate, so an example is an inhibitor binding, right? What happens is that the inhibitor might have very high affinity to the enzyme that it does not turn over into product. That very last step of the reaction does not take place. So if the inhibitor has a higher affinity to the enzyme, then the substrate cannot bind to the enzyme, or the enzyme inhibitor complex does not, turn over into product. So we're going to be talking about those really on our next chapter. But meanwhile, that specificity of the enzyme has to do, or the lowering of the energy of activation has to do with how the enzyme stabilizes the transition state, or the enzyme substrate complex. All right, so I'm going to erase that because that comes later. All right, so there are a number of different mechanisms that a given enzyme uses to turn over substrates into products, right? And one example is acid-base catalysis. We talked about the side chains of the amino acids and their pKa values, and we could think about which ones can be involved in acid-base catalysis, right? Ones that can protonate and deprotonate. And in that acid-base catalysis, so it's proton transfer. Another example is covalent catalysis. And just like the name implies, we're talking about a given enzyme that during that, turnover of the substrate into product, the enzyme catalyzes it. The mechanism of that, process that is taking place involves covalent bond formation and nucleophilic catalysis, right? And then we have metal ion catalysis, in which metal ions positively charged, so that's an example of an electrophilic type of catalyst. And then we have the proximity and orientation effects, right? The efficiency and the specificity of a given enzyme to a given substrate. And then we have the binding at the transition state, forming the enzyme substrate complex, for instance, right? That high affinity that increases the concentration of the enzyme substrate complex and its turnover into products. Okay, ta-da-da. Let's move. So, if we think about biological reactions in general, they are all susceptible to proton transfer or to acid-base, or have an acidic or basic groups, right? And can be involved in acid-base catalysis. And if we think about the side chain of amino acid residues and how those are influenced by changes in pH, then we know that those can also be involved in acid-base catalysis. If you think about the side chain of aspartic acid or glutamic acid, right, which have pKa of about 3.5 or 4. You have histidine that has a pKa of 6, cysteine that forms disulfide bridges, and we have tyrosine. Cysteine is about 8, tyrosine is about 10. Then we have lysine that is about 10.5. All right. So if you think about the catalytic activity of an enzyme, right, it is sensitive to pH changes. If you have any of those amino acid residues at the active site, changing the pH of the solution will change their ionization, right? It will also change the overall folding of the protein or enzyme. So one of the experiments that we usually do when measuring the activity of a given enzyme is what we call a pH profile, right? We are trying to find the optimum activity of an enzyme with respect to pH changes, right? So suppose this is a reaction in the overall rate of the reaction, right? This is the reaction rate, right? So you have the enzyme, you add the substrate to it, it's forming an enzyme substrate complex, and then you have the enzyme and the product, and you're measuring the rate of the reaction. Or in other words, you're measuring the turnover of that substrate into product, right? So you're measuring the rate with respect to pH changes. So that's the rate, and that's the pH changes. Why pH changes? Because as the pH is altered, the overall charge of the ionizable groups will change. The overall folding of the protein might be affected. And then you're measuring the rate at pH 5, and then you measure the rate of the reaction at, let's say, 5.5, and then you measure it at 6, and then you measure it at 6.5, and then you measure it at 6.8, and then at 7, and then 7.5, and then 8, and then at 9, right? So based on that, this is what we call a pH profile. We know that the optimum activity of the enzyme is around neutral pH, right? So it varies, but most are around neutral pH. And then if we're running a series of reactions, we know that we want the reaction conditions to be at the optimal pH of that given enzyme. All right, and these are really just examples of a general acid catalyst and a general base catalyst. And you have seen keto-enol tautomerization reactions in organic chemistry, but if you have a keto group, you have an acid, right? In the transition state, where bonds are formed, is just really indicating what bonds are formed and bonds are broken. You have this proton, you have something that is taking the proton, right? It's not going to dissociate on its own, but then you have an enol. And keto-enol tautomers are very common, right? Kind of switches between both. In a general base catalyst, you have a base that is deprotonating, you have a carbonyl oxygen, those electrons are going to the oxygen atom. Not sure what you guys see or don't see, but then this bond is broken, a bond is formed, a bond is broken, and then you have a carbon with an oxygen. We know we can move electrons with the carbonyl oxygen, and then we have an acid that is donating a proton, and then we get the final product, right? So in general, acid catalyst, we have an acid, right, is donating a proton. General base catalysis, we have a base that is accepting a proton. But you always, in acid-base reactions, you'll need an acid and you'll need a base anyways, right? Something is donating a proton and something else is accepting a proton. So I think it's straightforward. An example of an acid-base catalyst is that of RNase A, which is a digestive enzyme that hydrolyzes different RNA components into their nucleotides, right? And it forms nucleotides, cyclic nucleotides in the reaction intermediate. And if you take a look at the tertiary structure of that protein, you'll notice some beta sheets. So that arrow is indicating the direction. It is going from the N terminus to the C terminus. And then if you notice, you have a small loop. And after that small loop, you have the rest of the peptide. And it looks like it's ending right here. So that's either the N or the C terminus. And if you notice, there's disulfide bonds that are stabilizing part of the protein. You have other disulfide bonds right here. But that's the active site where the substrate will bind and be converted into products. And we're going to look at it, right? But that's just the overall tertiary structure of the protein. Okay. So what exactly is happening? It's an acid-based catalyst, and one of the most commonly amino acids that you'll see involved in acid-base catalysis is really histidine, because it has a pKa value near physiological conditions. Its pKa value is about 6. Now, you have to also keep in mind that that pKa value depends on the overall folded protein and what we call the microenvironment. So it varies slightly from 6. It's not exactly always 6.04. That is true for all the amino acids. All right, so this is really denoting the amino acids around the active site of the protein, all right? So think about it as the active site pocket, or that pocket near the active site. And then there are 2 amino acids right at the active site, and those are histidines, right? That's histidine 12. I'm just gonna call it, actually, let's just in case you don't see the actual figure. So this is histidine 12, and nearby the active site is another histidine residue. But that particular histidine residue is actually protonated, right? Well, we know that histidine, and it's histidine 119, and that again goes back to the overall protein, right? Or you can have, or enzyme or polypeptide, where you have amino acids that are far apart in sequence. One is at position 12 of the sequence, and one is at position 119 of the sequence. And in a folded protein, they are near one another, right? So, okay. So, they're both histidine residues, and both histidine residues have the same exact pKa value. So why is one histidine initially protonated and the other histidine deprotonated? Again, that has to do with the microenvironment of that folded protein. So what happens is histidine 12 will initially act as a base, right? And what RNase A does, it cleaves RNA, right? It's going to cleave a phosphodiester bond. No, it's going to cleave, a phosphodiester bond that is joining two sugars in this case, right? But it's really the phosphodiester bond within the backbone of an RNA molecule. And we haven't really talked about those yet, but we will at a later chapter, right? That's the 3'. Phosphate. Space. I need space. All right. Yeah, it's kind of tough with space. So, all right, and here's another phosphate, etc., right? O minus and double O and O. So it's really hydrolyzing that particular peptide bond. And what happens is that initially one of the histidine 12, is acting as a base, and it's deprotonating the hydroxide, the 2' carbon, right? And those electrons. And now that oxygen has a lone pair of electron, right? Typically a bond is formed, a bond is broken, but what's happening is that those 2 electrons are going in and attacking the phosphate. And now this bond is cleaving, whereby those 2 electrons are going in and deprotonating that particular histidine residue. So what we get is that, the histidine at position 12 is acting as a base initially, all right? And it's promoting that nucleophilic attack between that hydroxide bond to attack the phosphate and promote that bond cleavage. So, now what is breaking off? A water molecule is needed because it's going to protonate this oxygen. Okay, so, and then what happens is now all of this is gone, right? This is what dissociates. And what we have left is this 2', 3' cyclic nucleotide. And it is a 2', 3', because when we're naming the sugar, this is the 1', this is the 2', this is the 3'. So it's a 2', 3' cyclic nucleotide, right? That is the intermediate. And then we need a water molecule, right? Now the water molecule now, if you notice, the histidine that was acting as an acid is now acting as a base, and it's protonating. And the histidine that was initially acting as an acid, as a base, is now acting as an acid. All right, so what happens at the end of the reaction is that the histidine residue that was acting as an acid becomes a base, and the histidine residue that was acting as a base acts as an acid. And at the end of the reaction, the enzyme goes back to its original protonation state, where histidine 119 is protonated and histidine 12 is deprotonated. Okay, again, let's just go over it really quickly once again. You have an RNA molecule, and you have 2 histidines at the active site. One is acting initially as a base, and the other one is acting as an acid, right? And that will cleave that phosphodiester bond between 2, within the RNA molecule. And then what you get is that basically this entire thing is, part of it is gone. Think about it like peptides, cleaving a peptide bond, right? Part of it that entire sequence dissociates. And then what happens is the histidine that was acting as a base initially is now acting as an acid, deprotonating, and the histidine that was acting as an acid initially is now acting as a base. And a reaction intermediate is that 2', 3' cyclic nucleotide, and then the final product is formed, and the active site of the enzyme goes back to its original protonation state. So that's an example of an acid-base catalysis of an enzyme catalyzed reaction. Some are covalent catalysis, right? So you have an amine. An amine is attacking a carbonyl oxygen, which we know is reactive, whether it's an aldehyde or a ketone. Then you form a product. We can have tautomerization reaction and form an amine or a Schiff base, right? It's accelerating the rate of the reaction by forming this transient substrate complex covalent bond, right? Okay, that's an example of covalent catalysis, or the conversion of acetoacetate into acetone, right? And again, going through, going through a transient catalyst substrate, right? So that transition state speeds up that reaction that is taking place. All right. All right. So if you think about a covalent catalyst, the other one had like an amine. Those have to be good nucleophiles. They also have to be good leaving groups. So they have to be able to dissociate at the end so that the final product is formed, right? And if we think about the side chain of amino acids, those are ones with highly polarizable side chains. Histidine, lysine, serine can be involved in covalent catalysis. We have a aspartic acid or cysteine, right? I think I'm over my time. All right. And here's just another example of them and what the nucleophile would be in that particular case.